So welcome back, students. Am I audible to all?
Yes, sir.
Yes, sir.
So now number is significant, so we can start. In our previous lecture, we were talking about the basis of means the main purpose of this module. As I mentioned, the module contained 2-3 techniques in which it's counts the hybridization or simply blotting, northern, southern, western. So the main purpose of any of these techniques is the detection. And then when you're talking about the detection, the main basis of detection, we talked that it is a complementarity. So you may ask that, sir, how in case of protein it can be a complementary thing. So for protein complementary, we will talk about when I'm going to, and I will discuss the western blotting. So before the two type of blotting, that northern and the southern and the northern, they both believe on the concept of your complementarity, that is for the your GC and AC, GC and AT, that's the Watson-Crick base pairing. And that's why we also talk about that two strand RNA is single stranded because we don't have the complementary strand. That's why it is single stranded. Otherwise, wherever it found a chance of being double stranded, it forms double stranded form. And so the main basis we mentioned that detection is the term probing, and this term probe is same as you use in case of your criminal means your criminal also investigation type of things. So probe simply means the finding, or the probe is a tool that you use to find. Okay, so in case of the molecular biology and in case of recombinant DNA technology, or wherever you are going to use the DNA, we use the term probe that is made up of your DNA or RNA. And then come to its length is around 100 to 10,000 base pair base long, depending upon what specific sequence you want to detect. And so detection is that we also talk about that detection that you are going to detect. That depends on the labeling, that label present over it. And label can be of 2-3 types. So that is one is the radioactive one. So your DNA have phosphorus, carbon, and hydrogen. You do not have sulfur. So if anything which is there, sulfur is there. For example, that is a protein. So protein have sulfur. So in that case, the respective probe, your respective label you can use. So your nucleotides are going to be labeled with any of these radioactive atom. So if this radioactive atom is going to be present in your this in your chain or in your probe, then you can detect the presence of probe. So wherever probe is going to be there, you can say that my known sequence, the sequence that I'm searching, is present there. So similarly, you can label with the fluorescence. So different fluorescent tags are there. That could see is the one example, and different other tags you all have studied in your chemistry, photochemistry. So the fluorescence tag that was there in your fluorescent photochemistry, the same all you can use here also. And then come to the primers. So primers we do not utilize for the purpose of as I mentioned in case of the hybridization, southern or northern. But primers we utilize for your PCR. There you are going to amplify a segment, and amplifying a segment means that give you the piece of product. So whether the sequence is present in there, so primer you are going to use as a marker. So probe is also a marker, primer is also a marker. So property wise, probe and primers both are DNA stretch and 5' to 3' sequence. They are having a specific thing with the primer is that being primer is enrolled in your application. So for that, it must have a 3' OH. Probe may have or may not have it. It doesn't bother to us, but for primer, 3' OH is must. And chain length difference is also there. Probe are relatively larger in size, whereas primers are relatively smaller in size. Their chain length is going to be around 18 to 24 base pair long, not more than that. Okay, so then we talked about the hybridization. So hybridization is the formation of duplex. So this duplex formation can occur only if your strand is going to be single-stranded, double-stranded DNA. They are already in duplex form, so they cannot form a chimeric duplex. So here, that hybridization or the duplex formation that we are referring to the hybridization of your probe and your complementary sequence present on your DNA sample. So whether your DNA sample is going to have a red sequence or not, that you are going to use. So DNA, RNA, and DNA, RNA, that type of the hybridization you can do. So annealing term UV is used the hybridization in case of primer. But generally in the hybridization also you can use the term annealing. So annealing means your DNA sequence that is going to be single-stranded goes and form a complementary base pair and got anneal. Anneal simply means it got stick to it. That is the annealing. Joining is called the annealing. And also we talked about this property that how double-stranded denaturation and renaturation occur. So before hybridization, it is must to denature it. Otherwise, you do not form the duplex. And then after denaturation, we are going for a specific renaturation. Means your probe should go and bind to the location. And we also talk about the non-specificness, how you can reduce. So by increasing the temperature, we can do that. Okay. So then we're talking about the solid sport. And solid sport, I mentioned that solid sport can be of two types, whether it is going to be membrane or it is going to be a silica or a glass slide. If it is going to be membrane, then we call southern, northern, or western, or dot blot. So these are different types of things. Wherever in case of the silica glass slide, we call it as a in tissue hybridization, in situ hybridization. Simple example is fish, where you hybridize your probe with the chromosome. Okay, and this hybridization utilized for the detection of syndromes also, like Down syndrome, Klinefelter syndrome, etc. So this type we do. Microarray is also type of hybridization, where you have a vast array of probes to which you add your DNA, whether any probe having the complementary sequence with your DNA, sequencing DNA, then that may bind to it. So you perform it in this way. Then we talked about how this blotting is to be done. So for blotting, we need a transfer system, and transfer system consists of a series of your filters, or you can say blotting sheets. So that blot simply means absorbing. So here sheets, they are absorbing at the migration from your migrated DNA occur using a capillary action, or you can enhance it through using the electrophoresis also. Okay, so now we are at the southern blot. So southern blotting is a procedure of transferring denatured DNA from agarose gel to the solid spore filter, where it can hybridize with complementary nucleic acid probe. Okay, so this if we are performing this thing, then we are going to call it as a southern blot. The simple thing is that first prerequisite is the agarose gel electrophoresis. So if you are running your DNA, if you have to blot your DNA, you have to transfer your DNA, and that technology that for the transfer you have to utilize a southern blot, then first you have to do agarose gel electrophoresis. Okay, so this electrophoresis is the first primary step, and even so there are some issues in this transfer after that. If your DNA size is going to be very large in size, then the transfer is not easy, because a large bulky DNA won't go and attach to your your membrane. So in that case, we do one thing: we make it, we cut it, we make it fragments. So fragmentation simply means restriction digestion. So restriction is going to have a specific sites, so they are going to cut at a specific sites, not at the random site, and it will not produce final. It will not degrade your DNA as a that you are going to get a final nucleotide. So you are going to have to fragment it, and fragment should be of a known size and a known location, not no random location. We don't want a random location; we want a specific location. So for that, we are going to utilize restriction and nuclease also in this process. So now if we talk about the how to perform it, so there's just a representation. So first process that we have to do first is the DNA extraction. You must have a DNA sample with you which you want to use for the blotting to check whether the DNA is there or not. This blotting we utilize for checking or cloning also. Like if you want to make a transgenic plant, in the transgenic plant you want to verify whether this DNA, whether this your gene has got incorporated within your genome or not. So for that you need to perform a certain blot. Okay, so first thing is the DNA extraction. So once you extracted the DNA, next task is going for the restriction digestion. So restriction digestion, if you take the DNA, treat with the restriction digestion, it is going to digest into different fragments. So once the fragments forms, now next job is to separate them, because all the fragments they are going to present together in your sample. So in the mixture, it is not easy to diagnose whether where your fragment is going to be, whether it is there or not. You can do in your mixture also. For that we are going to utilize a term dot blot, but that is not going to be not going to help you in the identifying or isolating the gene. So that's why the southern blot is a more specific thing. It tells you clearly what size of the fragment is going to have your DNA sequence, and at that site what other restricted nucleases are present. Okay, then since only single-stranded DNA bind to the filter, DNA must be denatured. So this is one thing, as I mentioned. Only single-stranded can bind to filters, and also can bind to your probe. So that's why you need to denature it. So denaturing steps to be done within your agarose gel. So to that agarose gel, we have to denature it. So denaturing, one is the temperature help to denature. Another one is the same as used in case of your plasmid extraction, that is your NaOH or alkali denaturation. So in presence of alkali, we denature it, and after denaturing, your strand DNA becomes single-stranded. Now this single-stranded form of DNA we have to transfer to the your filter paper or your membrane. So this transfer we generally utilize a capillary flow. With the capillary action, solution is going to move from down to up, as in case of your in your paper chromatography, as your sample is also moving up. So here also in this case, transfer with the capillary action, DNA also move to our upper upside, and it will reach to the your filter membrane, and there it is going to you for the hybridization. So hybridization simply means that you are adding your probe weight. So probe, if you are adding, then probe will go and bind. The binding of probe you have to visualize. So visualization, for that it must be labeled. So once you label it, then you can detect it. This is a simple probe brief. Okay. So here is a flow chart how to see it. So here you can see that your DNA is there. So large when you digest it, large fragments are going to be there. So these fragments are going to be of different size. So these are different size fragments you have to arrange upon the basis of their size. So then you have to jump for electrophoresis, restriction digestion, then electrophoresis. Electrophoresis on the average will separate you and arrange you on the basis of decreasing order of your size. Now you have to denature it. So denature means you have to treat it with NaOH. And now the single-stranded one, now you have to transfer to the membrane. So membrane to have transferred the single-stranded one. Now the single-stranded is there. To this you have to add the probe. So this is a probe. So probe where it found the complementary sequence. Like in this one, it found with 3 complementary sequence. Wherever in this it found just 2 complementary sequence. So depending on wherever the complementary sequence is present, you can detect. So when you visualize it, it is a fluorescing one, and it is going to fluoresce. So fluorescence 2 signal going to be there in this case. If it is a radioactive, then similarly 3 signal going to be there. Okay, so here you can see in the right panel, these black dots are there. All these black dots are of your bands, and they are on a photographic plate, and how these different probes you have used. So this is how the southern probe usually perform. So now, as it is a very large module is there, so we need to discuss a bit about what methods we are going to apply, how really we are going to perform our experiment. First thing, as I mentioned, restriction endonuclease are used to cut high molecular weight DNA into smaller fragments. The DNA fragments are then electrophores on the hydrogen electrophores, which are taken by size, as we already have shown there. If some DNA fragments are larger than 15 kb, so this must not that why the main thing is what the main issue is there. Then prior to bloating, the gel may be treated with the acids. So thing is that if your DNA fragment is going to be a bit larger in size, then that make difficult because it's a capillary action they are moving up, and larger size they won't move easily. So for that you have to use a smaller size. So if your fragment, because the restriction endonuclease is for we are treating with, then what happens? Then thing is that in that case you can't say that the fragments are of this size only you are going to get. Sometimes you may get a larger fragment also depending on whether the residual site of that enzyme is present in that your gene sample or not. So in that case, gel may be treated with the acid. So if you treat your gel with the acid, but it may depurinate the DNA fragment and break your DNA into smaller pieces. So it do in a way that your DNA strands they remain near to each other. They want to migrate uh, longer. So alkaline transfer methods are used. So alkaline methods transfer simply means you're transferring the presence of alkali. The role of alkali is denaturing it, denature double-stranded DNA to the single-stranded form. Okay, so the denaturation in alkaline condition may also improve the binding of negatively charged thymine residue of DNA to the positively charged amino. So why these two DNA is going to bound to the membrane? So because membrane contains the amino group, which is a protein. It's amino acid, amino groups are there. So it's a nitrocellulose membrane, so nitro group is there. And so this nitro group, to this nitro group or amino group, if they're there, sorry, amino nitro is going to like. Okay, so in that way, these they help in the interaction and they help in the binding. Okay. Separating into single-stranded DNA, later hybridization probe, and destroy any residual RNA that may still be present in the DNA. The choice of alkaline over natural transfer, however, is often empirical and may result in the equivalent results. So it's not just always that you cannot go for a neutral transfer or a way, but it helps you in it makes you better. Okay, so then, as I mentioned, a sheet of nitrocellulose, alternative nylon membrane also you can use, or PVDF membrane I mentioned that also you can use. Then what to do? You apply pressure evenly on the gel. When you apply pressure on the gel, so applying a pressure facilitate liquid to migrate. So gel is a contain liquid, but gel is not a solid; it's a semi-solid. So when you apply a pressure, then its liquid portion start migrating upward. So this migration, this pressure, will help you in the migration of your DNA from your gel to the membrane. Okay, so this pressure is going to help in this way, and the membrane is then baked in vacuum. So now once the transfer is over, then you have to go for a baking step. Means when the transfer is done, now that you have to bake it. So baking means you are heating at 80 degrees Celsius for 2 hours. So why you are baking it and doing, or you can expose to ultraviolet radiation. Also, the main purpose is, as we mentioned, that interaction between the DNA and membrane is there. With that interaction, your DNA is attaching to the membrane, but that interaction is not permanent. It may leave your membrane also to make permanently attached. We make it to cross link. Cross links mean we want to make a hydrogen. We want to make covalent bond between nitrocellulose membrane and your DNA. So For this cross-linking, we treat it with the UV radiation, or we are going to heat it. So at higher temperature and the UV radiation, they make the cross-linking of DNA with the membrane. Then the membrane is exposed to hybridization probe. So now you have all these things crossed. You have denatured it. So your transfer is in the alkaline condition. So it transfer in the single stranded form. Then you cross-linked also. Because once you cross-linked and denatured, whether you are now it's do not form double stranded immediately, or we keep a condition also a bit alkaline in our process. Okay. So then membrane, you are going to transfer the hybridization probe. So these are the your single-stranded DNA which having the non-sequence to you, and if this non-sequence is going to bind or not, that depends upon the presence and absence. So this DNA, the probe DNA, is labeled so that it can be detected. So usually by infrared radioactive tagging or molecule with a fluorescent or a chromogenic dye. So the main purpose is of your radioactive label, fluorescence or chromogenic dye is that when the probe is going to bind, you can visualize it, because as I mentioned earlier also, DNA you can't see with your eyes, so you can see probe also, but you want to see that whether your probe bound with it or not. So wherever your probe has bound, so you want to see a distinction between them. Your signal should come from your probe only, not from the other DNA, so that you can tell that this sequence is present in this size and that size of your band, and this sequence is present 3 times, 4 times, 5 times, how about this many number of times in your DNA. So if it is going to be radio labeled, then you have to need an X-ray film. So place our X-ray film and visualize it. So autoradiography is being done. So it is auto radiation. You need not to do anything. Just place your gel or your membrane over the X-ray film. It automatically gives the impression. And for the fluorescence, you definitely need excitation and emission radiations. And for chromogenic substrate detection, if it is an enzyme, then you need to add a chromogenic substrate with it. Or if it is already colored thing, then also you need to do it. Okay, so this is a graphical representation how you have to do. So the first step, you can see the gel is there. So over the gel, you have to place a nitrocellulose filter. So as you place the filter, then you have to put a blotting sheet over it. So these blotting sheets are there going to help in the capillary action, and with that you have to over it you have to put a weight, and then DNA is being transferred. So as you transfer, you have to remove the membrane, and then after the membrane you have to add the DNA probe. So when the DNA probe is going to bind wherever it do, then you have to wash it. So washing is a must. If you are not going to wash your probe, then randomly, non-specifically, your probe may be present, then it may give the signal whether it is binding to your DNA or not. It may give the signal. So to avoid a false positive result, we have to wash it thoroughly so that there is no residual probe is remaining in your sample. So once you have washed it, then expose X-ray filter to your nitrocellulose membrane where you have transferred, and then it will give you impression over your membrane that these are the fragments where you are having uh, your probe. So this is a simple sort of way of detection, and you are going to utilize it. So more details, I don't think you need to go, and I'm not discussing these also. How to prepare the gel preparation for the blotting. If you need to read these things, I will share you these slides also, where you can get a detail of your southern blotting. Now, this is the main thing that I want to mention, the how to label your probe. So the labeling of probe is to be done by the enzyme that you know, the polymerase. So add 39 ml of designed probe to 5 ml of primer mix. Denature for 5 minutes, 99 degrees Celsius, and keep the mixture on ice. Now label buffer, add labeling buffer containing DNTPs with 2.5 ml of 32P. So there must be phosphorus, it should be P32, and 2 ml of PNO fragment. So this clean off fragment is polymerase enzyme, and it is going to label your DNA, because here you are not going for a polymerase reaction. So just want to add. So this clean fragment have both the activity. It have a 5' 3' polymerase and 3' 5' nuclease activity. So it also proofread while it is replicating again. So once clean fragment complete its task, your strand is going to be labeled. So then you can denature it and separate the double stranded. So now single stranded is going to work as a prop. Incubate your mixture to allow the radio level for 10 minutes to 16 minutes at 37 degrees Celsius. So the radio labeling is done by the cleano fragment. So as we already, when we're talking about the enzymes, that time also we mentioned that cleano fragment is going to be used for the purpose of radio labeling. Okay. This is again the what are the different buffers, what are the different recipe you need to use. So most important thing you should and you need that you should first need to denature your probe also before putting it, because when you label your probe, it also becomes double stranded. So you need to denature it and then to add. So blot washing, this is the most important factor as mentioned. So washing is going to remove a non-specific thing, any unwanted things that may be in case of your DNA, or it may be impurity. So all these things you can remove by washing. Then the autoradiography is simple autoradiography. You have to place your X-ray film over the top, making a sandwich. So when you do the sandwich, just in this thing you need to perform in the dark room, because if you open your X-ray film open, then your X-ray film may got damaged by the radiation present in your visible light also. So then place at 70 degrees Celsius for 40 hours before developing the film. So then this is the way how to perform. Then come to the precautions. So what precaution you need to do while performing southern blotting experiment? First simple thing is wear gloves and lab coats while handling the southern blotting materials. Make sure no air bubble should come and trap in the gel, because as I mentioned while I talking about gel electrophoresis, if any bubble is there, then bubble is going to break the paths. And if path is going to break, the capillary action is also going to be break. So transfer is not going to be an efficient transfer. So for that you need to should we avoid the any air bubble between the your membrane and your gel. Cotton paper that may cause DNA run irregularly, and monitor the agarose while heating on the left, as it is liable to boil quickly. So you can't heat agarose your DNA so much that it may melt. So gently remove the gel foam and to avoid the collapse. So these are all the that you need for preparing your gel. It's not specific for your southern blotting. Slowly release the sample into the well. This is also for the smooth transfer. Once centrifugal memory is placed, don't move it at DNA transfer may already have taken place. So once you have placed nitrocellular membrane over gel, now you can't do in a way that you can keep on moving it. Because what happens? So once you placed over the gel DNA, because whatever DNA that is present on the surface, that have already started the migration to the your nitrocellular membrane. So once you fix it, so it is a take a proper precaution for placing over the gel. So now coming to what are the strength and the limitation of your southern blotting. Southern blotting is a retrieval technique to analyze long stretch of DNA such as multi-kilo base restriction fragments. So if restriction fragments are a bit larger in size that you can't analyze using cloning or other things. So even before go for the cloning, you need to check whether your sample is going to have your gene of interest or not. So for that you need to go for the southern blotting. The expression level and the expression profile are better detected as compared to the other genomic analysis method. Southern blotting had wide area of application in the field DNA fingerprinting, including paternity testing, maternity testing, forensic studies, person identification. So all these purposes you can utilize the southern blotting. The method is cheaper than genome sequencing. So where you think that you don't need to go for the genome sequencing, you can simply having a probe and you can check whether the DNA is present or not. So simply you need to go for the genome sequencing. So genome sequencing is a little bit expensive one. So in that compare it is a bit cheaper. The method may require a large quantity of DNA, so this is a limitation. You can say that you need a large quantity of DNA for sequencing, and PCR you don't need a large amount of DNA, but here you need a large amount of DNA. And the quality should also be there. You can't compromise with the quality as compared to other genome analyzing techniques. So these are the few things that you need to check while performing southern blotting. So here are some solution that might be required while you're performing. So these all things you need to do. You need to add first thing is HCl. That HCl you need for depurinating or breaking if your cell is going to be a bit larger in size. Denaturing solution contain NaOH, so that's why denaturing neutralizing solution. So for neutralizing you need a base, which is a neutralizing solution. You need STS for washing. You need a SCS, which is sodium chloride, sodium citrate solution. You need a pre-hybridization buffer, so these are mixture of SSC and STS and so on. Then hybridization buffer, which is going to allow you the hybridization. So here the temperature should be a bit higher so that non-specific binding should not be there. Blocking reagents, so to block, because once the transfer is there, so for that you want to avoid a non-specific binding, because as we mentioned that transfer of DNA over the membrane is because of membrane have an interaction with your DNA. So if you are going to add probe to the nitrocellulose membrane, then chances are there that your probe will also bind to the membrane surface here and there. So we do not want that DNA probe will bind here and there, or it will not specifically with the ionic interaction, nitrocellulose membrane may absorb your probe. So for that, what we do, we block. So blocking is a necessary process. So before going for hybridization, we first have to block. So blocking is done by DNA that we call the horse-form DNA, so that that is going to bind to throughout your membrane, except where DNA, your DNA or your sample of interest already bound. All other places your sperm DNA is going to bind, and it will block your membrane. So now that all the sites of your membrane are blocked, so in that case your DNA, in that case your probe will not go here and there and will not bind non-specifically. So this is a simple thing that you have to do in case of this is the role of your blocking agent. And next is malic acid buffer. This malic acid buffer also is to facilitate your transfer. So this is it's a brief thing of southern blotting. So if you have any question over the southern blotting, please ask. Have you any question from the southern blotting?
Sir, is there any other way to do the denaturation? Because usually don't we just heat for denaturation?
Yeah, alkaline denaturation we are doing now. We are not doing a heat denaturation. We are doing alkaline denaturation. Because if you do the heat denaturation, for the heat denaturation you need a temperature of 95 degree or 100 degrees Celsius. Okay. So that temperature is going to melt your gel. So we can't do heat denaturation in this case. We can do only alkaline denaturation or chemical denaturation.
Okay, thank you, sir.
NaOH is going to use for the denaturation, not the heat, because heat is you are going to use for cross-linking, not for the denaturation. Cross-linking of your DNA to the membrane. For that you are heating. You are heating membrane, not the gel. So after the transfer you are heating, not before the transfer.
Okay, thank you, sir.
Any other? Okay, so if no question, then we have to conclude today a bit early, because today is an orientation program also. So for that also I have to join. So please turn on your camera when I call your name. Adish. Adish.
Present, sir.
Atoiba. Ayushi,
Present, sir.
Demetrius,
Present, sir.
Demetrius,
Present, sir.
Atharva, yours done.
Hello?
Demetrius, yeah.
Present,
Devanshi,
sir.
done. Ishita,
Present, sir.
Ishita,
Present. Present, sir.
Anki,
Present, sir.
Kshitij,
Present, sir.
Kshitij, Manaswini,
Present, sir.
Namrata,
Present, sir.
Nandini, Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba,
Present, sir.
Anjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tanvi,
Present, sir.
Dev, Vivek, Vivek,
Present, sir.
Yuvraj, Yuvraj,
Present, sir.
and Demetrius and Nandini.
Present, sir.
Sir, I'm present.
Turn on your camera also, Nandini and Demetrius.
Sir, on it.
Sir, this is
Okay, so thank you all. Stay safe, stay happy.
Sir,
See you
Naveen
again
sir.
on Monday. And on Monday we'll talk about
Sir, excuse me.
yes.
Sir, Vivek here. I'm present.
Vivek, speak a bit loudly. Your voice is not coming.
Sir, I'm saying I'm present.
Yeah, yours done. I didn't call your name again. I just called Demetrius and.
Okay, sir.
Okay, so thank you all. Stay safe, stay happy.
Thank you, sir.
Thank you, sir.
Hello. कॉल तो करते हैं पर मुझे कॉल कर दो